Assalamualaikum warahmatullahi wabarakatuh. Today I will present about the method of DNA vaccine delivery and how it works from Group PBL 21 and 22. There are three types delivery of DNA vaccine, which are parenteral, topical applications, and alternative method. For the parenteral, it can be administered with the select injection or gene gun. For the select injection, it will use a standard hypodermic needle and gene gun delivery. It given intramuscularly in the skeletal muscles or intradermally, and it will deliver the DNA to the extracellular spaces. Select injection also assisted by the electroporation that will be explained in the next slide. Immune response also can be affected by the various factors, including the needle type, needle alignment, speed of the injection, volume of the injection, muscle type, age, size, and physiological conditions of the recipient. So, what is electroporation? Electroporation is when the electric field passes into the tissue and form the transient pores in the cells. It will trap the DNA vaccine into the cells. This procedure rapid, simple, and efficient. It also will increase the cellular and immune response. So what happens is that the DNA vaccine will be injected into the selected muscles or skin tissue. There will be electropulses applied at the needle of electrolytes, which then will form the electric fields. So what electric field do? It will create temporary opening in the cell membrane that allow the DNA vaccine to enter the cells. Then the cell membrane will receive back and the trapped DNA cause the cell to produce the antigen that coded by the DNA. APC will engulf the antigens and carry them to the lymph node. So what happens in the lymph node? There will be interaction between the APC and other immune cells that result into the productions of the antibody that will prevent the future infection. Next, under the parenteral is gene gun. It accelerates the plasmid DNA, pDNA, that has been absorbed onto gold or tungsten microparticles into the target cells by using compressed helium as the accelerant. It also delivers small amount of DNA. The target cells, ED, abdominal skin, vaginal mucosa, surgically exposed muscles, and other organs. The next method to deliver DNA vaccine is topical application. Our target tissue in topical application is ocular or intravaginal. This method is simple, painless, and cost-effective. The level of immune response is relatively low. The last method is alternative method. There are many alternative methods, include mucosal surface delivery, cationic liposome given intramuscularly, biodegradable microsphere given intravenously, it transfect the tissues systematically, recombinant adenovirus vector given intraperitoneal, attenuated Shigella vector for the oral immunization to the intestinal mucosa, aerosolized cationic lipid formulation for the nasal or lung mucosal membrane. Next, I will be explaining about the mechanism of the DNA vaccine. First, the plasmid vector that expresses the protein of interest, for example, the viral protein, is under the control of appropriate promoter given to the host. After uptake of the plasmid, the protein is produced endogenously and intracellularly will be processed into the small antigenic peptides by the protease. The peptides then enter the endoplasmic reticulum by the membrane-associated transporter that will bind to the MHC class I molecules and presented on the cell surface. Subsequent CD8 plus cytotoxic T cells are stimulated and evoke the cell-mediated immunity. Next, the cytotoxic T cells will inhibit the virus's by cytolysis of infected cells or non-cytolysis mechanism by production of the cytokines. The foreign protein can also be presented by the MHC class II pathway by the APC that will elicit the helper T cells response. CD4 plus cells will recognize the peptide form from the exogenous protein that were phagocytized by the APC Then it will degrade to the peptide fragment and loaded onto the MHC class 2 molecules. Depends on the types of CD4 plus cells bind to the complex, it will stimulate the B cells and it will produce the antibody. So here the DNA vaccine will be injected to the host, and then in the host it will produce the protein that will be processed into small antigenic peptides by the protease. Then the peptides will enter the endoplasmic reticulum and bind to the MHC class 1 molecules, and then it will be presented on the cell surface. So it will cause the cytolytic T cells stimulated and will evoke the cell-mediated immunity. So at the same time, the viral protein also will be phagocyted by the APC and it will cause the fragmentations of the protein into the small peptide. And it will bind to the MHC class 1 molecules and it will cause the productions of the antibody by the B cells. These are the references that we use to prepare the slideshow. So that's all from us. Thank you.